All right. Our next and final presenter is Sally McFadden of the University of Newcastle. And Sally's going to walk us through how acoustical imaging is used to research and how it may someday help cure the ever-increasing cases of nearsightedness. Afterwards, we'll have a live Q&A.
Hi, my name is Sally McFadden. I'm located in Newcastle, which is about 2 hours north of Sydney. And I want to talk to you today about approaches that we've been using to try and solve the problem of short-sightedness. Now, this problem occurs when images, instead of being well focused at the back of the eye, they're actually, the eyes grown too big, too long, and so that images are short focused. And that means that we've got blur at the back of the eye. Now, traditionally what we do is we put negative spectacle lenses in front of the eye, which puts the image back on the retina, but it doesn't fix the problem of the fact that the eyes are actually too long. And in high myopia, where we've got a really elongated eyeball, then we end up with these blinding pathologies where the retina can detach, these bubbles occur in the back of the eye, in the skin of the eyeball called the sclera, and we also get profound blindness from basically the eyes, the cells in the eye, the visual sensing cells in the eye dying. The prevalence rates are alarming. Over the last few decades, we've seen enormous increases, and it's now predicted that by 2050, half of the world's population here will actually be myopic. And those with high myopia, it'll be something like 10% or nearly 1 billion people. And there's really no treatment for high myopia. And we've started to develop treatments for myopia, but there's nothing there for high myopia. And so what we've been able to show is that myopia can progress into high myopia and into the pathologies of that cause profound blindness. The early studies that were undertaken, and this occurs across species, were using high-frequency ultrasound in order to be able to extract non-invasively the structure, the distances within the eye, and calculate how much the eye is actually growing. And in these studies, what we found was that if we put a spectacle lens in front of the eye, it would cause the image to go behind the retina. As a consequence, the eye would actually accelerate its growth. And of course, that means the eyeball is now much bigger. If you take this lens away, you've got sort of an elongated eyeball, which is basically a myopic eyeball. Alternatively, the eye does the opposite when you put a positive lens in front of it, which short focuses the image, and now the eye actually inhibits its growth. And it's this signal, this inhibitory signal, that's been integrated now into spectacle lenses for children to try and put a signal in there that might slow down that excessive eye growth that's occurring. So we know that both light and space influence myopia. For example, the prevalence rates of myopia went up excessively during the COVID epidemic. But we also know that if children go outside, that it can protect them from developing myopia, whereas doing a lot of inside study, or for that matter, living in small homes, can induce greater amounts of myopia. So these factors interact. So basically, we know that what causes myopia is to do with visual signals. You can't blame your parents for your myopia. It's to do with what you're using your eyes for and what our children are using their eyes for, and that causes a complicated cascade which actually changes the structure of the skin of the eyeball, the sclera. And this phenomena occurs across different species, even in fish, and it's a phylogenetically ancient sort of conserved pathway that is designed to set the eye up so that it will actually be able to see what it is that you spend your time looking at. Of course, if we're not in a hunter-gatherer society, or if we're not out spearing animals, then always seeing up close is actually not desirable because you can't see things in the distance. So we've been using acoustic methods, such as scanning acoustic microscopy, to try and measure how weak that sclera has become. It's well known that it's become weakened, and the types of treatments that we're looking at are designed to try and strengthen this outer coat of the eyeball so that it won't end up getting those outpatchings, and so we won't end up with them.
Thanks for the overview, and on a very interesting and relative topic. We're all concerned about nearsightedness, it seems. And now let's dive into the questions for your session. The first one is pretty straightforward, and it's from Richard Lovett, and he just wants to know, do phones and computers cause myopia?
Well, there's certainly a correlation between how much study you do on a computer and how much myopia you will develop. But in reality, it's really unclear exactly what the visual signals are. And we know that it's something to do with the brightness of the screens, the wavelengths, which interact with the way the eye grows. So certainly there's a lot of acceptance in the community that there's something to do with screen technology that can interact with the way the eye is growing and set it off. But if you think about modern environments, it's the lighting in general in our modern environments, especially urban environments, that has changed over the last few decades. And this has influenced the way the eye grows. And we can show that in those animal models, that it does change the way myopia develops.
Oh, okay. Richard also pointed out that the last 30 seconds of the presentation seemed to cut out. I don't know if anyone else picked up on that. Any chance you can kind of provide us a couple talking points from that so we know what we missed?
Oh, yeah, sorry, I was just, sorry about that, but it was just talking about the treatments that we can apply to the outer skin of the eye in order to try and stop the development of myopia, especially high myopia. These treatments involve changing the actual structures within the sclera. It's a bit like Botoxing the eye, although it's not really Botoxing it, but it's to try and stiffen the structures there and literally change the sort of chemical composition within the sclera and put it back to normal, because it sort of becomes very loose and elastic during the development of myopia, and it becomes sloppy. And under the intraocular pressure, it expands basically. And that's one of the reasons why the eye becomes so elongated. And so what we're hoping to do is to be able to basically what we call cross-link the sclera in the right place at the right time in order to stop the development of myopia. And we have preclinical work in this space to show that that definitely does work. And so that's really what we're keen to develop in the coming period.
Great, thank you. And thanks for summing that up for us. I think we have one more question here from Carrie Sager. If fish also suffer from myopia, is there any indication that animals we have domesticated could also be myopic if confined to the house or just are indoors a lot?
Absolutely. I mean, this is a phylogenetically anciently evolved system. And if there's been studies showing that if you put animals in small environments, or if you make monkeys watch TV, for example, or have low ceilings, then they definitely develop myopia.
Gotcha. And we actually do have one more question from Richard. He wants to know two things. One, how do you stop that cross-linking that you mentioned? And can you clarify how this relates to acoustics a bit more?
Yeah, so in order to do the cross-linking, what we need to know is exactly what the biomechanical properties are of the sclera and how we've changed them. And so we need to measure it, basically. And so we're developing acoustic-based or ultrasound-based systems to be able to know exactly what the strength of the sclera is, so that we know that we haven't over-strengthened it or under-strengthened it, and that we're doing it in the right place within the eye. So we basically want to use, and what we're doing is using ultrasound in order to image the actual functional capacity of that skin of the eye. And so that's really needed for when we're doing that cross-linking.
Thanks so much, Sally. If everyone's okay with it, I think we still have time, and there are a couple more questions. So we have one that says, how do interventions like LASIK impact myopia?
So LASIK is a bit like permanently putting a contact lens on your eye. It's just changing the power of your cornea. Unfortunately, that doesn't do anything to the fact that your eyeball is too long. So if you're already myopic and you do that, it's not going to help. And it's also the case that correcting your vision doesn't stop the eye from still being, if you're still in a myogenic environment, if you're still subjecting your eye to the kind of light and close focus conditions that cause myopia, which in fact most urban environments do,
then it's not going to help having the LASIK operation. Certainly it'll allow you to see clearly initially, but if you're already myopic and your eyeball's too elongated, it's that elongated eyeball that causes the problems of the profound blindness.
Gotcha. Thanks, Sally. And I think we have just one more from Richard Lovett, and he's asking, well, first, this applies to childhood development, or do adults also produce or develop myopia by reading fine print, for example?
It's definitely occurs during childhood development from babies onwards. And so, you know, the big increases that we're seeing there in terms of the development of myopia are from the way our children are interacting with their environments. But having said that, because it's something that occurs while the eye is growing, and the eye grows, you know, prominently in youth and up into the teenage years, and it's still growing even up to the age of 25 or so, or 30. But after that, your eye size is pretty fixed. So, however, there is turnover of proteins and things within the eye, which can also be affected during later years. And there's very little work or study that's been done on non-growing eyes. All of the work in the animal models that we use are all under growing conditions. But actually, if you do start to restrict the type of vision that an older eye sees, you can still actually induce forms of myopia. But in general, this is it's the way we're raising our children that is causing these huge increases that we're seeing.
Certainly good to know. As we were chatting, one more question popped in, and I do believe we have time for it. So, Richard, you get one more in under the wire. So, this is another question about cross-linking. Is cross-linking done by using drugs or lasers or something else?
Okay, so there is a method that's on the market for cross-linking the cornea, and it uses a photo initiator that you put onto the surface of the cornea, and then you use light to activate this cross-linker. In that case, you use UVA light, which is pretty dangerous for the back of the eye. You wouldn't want to use UVA light. But there actually are many other forms of ways you can cross-link. There's cross-linkers that respond to visible light, which is quite safe, or to long wavelength light, which we're working with. But you can also cross-link with just straight chemicals. So there are chemical cross-linkers as well that people have been trialing, and we've also been working with some of these chemical cross-linkers. So there are multiple ways that you can change the structure of that skin of the eyeball and create these extra links between the collagen fibers so that they become less elastic, stiffer, if you like, and stronger.